Okay, so welcome back everyone. Welcome to the evening lecture. So it's my pleasure to introduce to you Kyunghyun Cho. So he's been with us in the summer school for some years now, and well, I guess many of you will be familiar with some of his work. So Cho did a PhD in Aalto University on a topic related to unsupervised deep learning. After that, so he was a postdoc at Montreal working on neural machine translation, and he's now an associate professor at New York University. So, well, besides academia, Cho also does consulting and scientific advising to several startups and NEOs. So, before he was a research scientist at Facebook, and well, he's now one of the co-founders of Prescient Design, so a company working within the Koch group, working on computational methods for drug discovery. So his talk here today is going to be addressing his work within Prescient Design, so not prompting today. I guess we're all excited with that. And well, thanks again, Cho, and let's go welcome the speaker.
Thanks for the very generous introduction. So you have all learned about supervised learning, unsupervised learning, how to deal with sequences. And I thought, I'm going to show you one particular application that is not in natural language processing nor in computer vision, where unsupervised learning, supervised learning, and sequence modeling all come together to do some wonders. And that is going to be on protein design. And in particular, at the very end, I'm going to talk a bit about the antibody design, to be more specific. So just to tell you what Prescient Design is, we founded Prescient Design January 2021 out of our research, or the 3-year-long research at NYU, as well as the Flatiron Institute run by the Samas Foundation, on how to use deep learning for biological sequence modeling. In particular, we focus on proteins. And we started to hire people, and then we started to talk with the various investors as well as the partners. And then one of the major partners that we were talking with was Generative. And Generative used to be the very first biotech in the world. It was founded in late 70s and was the first company to develop and commercialize the human insulin that was created at the lab. Before that, it was all about, let's take a pig and then trying to, get the insulin out of the pig and then hoping that that insulin was going to work on humans. But from, let's say, late 70s and early 80s, we could actually produce the human insulin, which is a protein as well. So we were talking with them, and then they told us to, scrap the United States funding round and just join Generative. So we joined Generative in August 2021. And then since then, we've been working as what we call Generative Accelerator, working on the proof of concept for what we call lab in the loop de novo antibody design platform. And we've been doing that. We have actually designed and tested several batches of antibodies already. And then I'll be happy to talk about that at the very end of my talk. So one of the major foundations behind the technology that we use in order to design antibodies, as well as any other types of the proteins or even a small molecules, is generative modeling. Or sometimes we refer to it as unsupervised learning, because in this paradigm, we work with a set of unlabeled examples only. So we're not working with any examples where we have annotations. And then when we think about generative modeling, you all have learned the various different types of the generative modeling algorithms or the unsupervised learning algorithms. For instance, yesterday you learned from Noah about HMM, which is also one of those unsupervised learning algorithms. There are always two extremely tightly related goals. One is scoring. We want to estimate an energy or score such that the energy assigned to a realistic looking examples are going to be always lower than the configurations that do not look realistic. And second goal is sampling. We should be able to, after training is done, we should be able to sample a realistic looking configuration from this trained generative model. Now, why are they tightly related? Because once we know how to compute or assign the energy values to the valid configurations, then we can always turn this score function into a distribution using what we often refer to as a Boltzmann-Gibbs formulation, or more often these days just softmax. Now that said, this does not actually imply that we can readily sample from this distribution defined using the softmax, using the score function coming out of these generative models. But if we try, we probably could to a certain degree. And what that means is that connecting this to a neural net simply means that we always build a very large scale neural network that's going to take as the input some configuration of the some configuration X, and then this neural net outputs just a single scalar. And this scalar can be really any value, positive, negative, whatnot. And then what we do is we train this neural net to maximize the log likelihood of data. Now, how do we often do that? You learned about the expectation maximization and various other, let's say, sophisticated techniques yesterday and day before and so on. But these days, what do? We just do the gradient descent. So we're going to write down the log likelihood of the data that is decomposed as the sum of the per example log probability across all the examples in the training set. And then we're trying to compute the gradient of this term. And then when you try to write the gradient of, let's say, individual term, what we end up with are two terms. First one is what we call positive phase, and the second one is a negative phase. And what does the positive phase refers to? The positive phase refers to the fact that we want to minimize the energy of an observed, so the positive example we have. So we're trying to ensure that the energy is going to be lower. Now we always want to increase the energy. That's how I normally think, but somehow these physicists have some wrong concept of the sign. So we always have to lower the energy of the positive examples. And the second term, the negative phase, tells us that we want to maximize the energy of probable example according to our current model. So it's like the whack-a-mole kind of situation. We say that, well, here are some positive examples. We want to ensure that the energy is going to go down on those positive examples. But on the examples, negative examples that according to our current model are really likely or are assigned already a low energy, we're trying to increase it. So this is an illustration of what happens. You can imagine that the data points corresponds to the black dots on the left side of the panel. And then the blue dots are the ones that are probable under our model. And then what we do is we're trying to lower the probabilities of all those blue dots while trying to increase the probabilities of the black dots so that we end up with this kind of, let's say, greenish, let's say, contour. So we can now train it using stochastic gradient descent, and that's exactly what everyone knows how to do these days, using tons of GPUs and whatnot. And then eventually, what we end up with is that this energy function is going to be lower if and only if the configuration X looks like one of the training examples in the training set D. And then all the other places, the energy value has to be really high. That is the goal there. And what does this mean? This actually tells us where the data is concentrated in this gigantic space of the input. So if I only look at all the configurations or the set or the subspace of the configurations over which the energy value is low, then I can see that there is going to be some kind of manifold where the data is concentrated. I'm going to use the term manifold extremely loosely here. However, you can imagine that this way of generative modeling is how we actually identify or characterize this low dimensional data manifold. And the data is always living in a low dimensional data manifold. Otherwise, we cannot really do machine learning. Only reason we are successful with any kind of machine learning technique so far is the fact that somehow the data that we work with or the problem that we work with lives in a much lower dimensional manifold than the actual, let's say, the seeming dimensions that we think they live on. So let's say we somehow train the generative model using stochastic gradient descent. And then what do? We want to sample from this generative model because we want to see what kind of things are considered likely that may not be part of the training set. And then you've been seeing all this, the DALI 2 and so on, we know how to sample, we think we know how to sample from it, but in principle, we actually don't, unfortunately. And there are many techniques such as, let's say, very naive, let's say, Gibbs sampling. Well, Gibbs sampling is not really naive, but let's say Gibbs sampling or the slightly more sophisticated, let's say, metropolis Hastings with a complicated, let's say, local proposal distribution. If our X, the input, is going to be continuous, and if our energy function was differentiable with respect to the input, we can rely on some of the dynamics. We can rely on the gradient in order to do a more efficient sampling. And of course, being a machine learning people, we should be able to train yet another machine learning model to sample from a given distribution as well. And then that's what that was the idea behind many of these advances in the line of the generative adversarial networks, which kind of, let's say, I guess, stopped advancing a couple of years ago, because I don't really see any paper on generative adversarial network anymore. They probably consumed all possible ways to name new generative adversarial networks. So, they are not writing the paper anymore. Anyway, okay. Unfortunately, none of these strategies tends to work well generally when X is a discrete and combinatorial. It's just that the space of X is so large, and also there is no inherent or the very natural metric structure that we can use in order to build a efficient sampler. So what do I mean by that? There's no, let's say, natural inherent, let's say, metric that we can use is that every point in this discrete combinatorial space tends to be pretty much equidistant away from all the others. So unless we put huge amount of prior knowledge, which is often not what we have, then there's no way for us to tell whether, let's say, one particular, let's say, word is more similar to that word or not. And that applies to everything beyond natural language as well. So in the case of, let's say, proteins or the antibodies, just looking at two different antibodies, or we're going to look at three of them, and then it's really impossible, nearly impossible to tell which ones are closer to each other than which others. And then that makes it really difficult for us to come up with a better sampler or even a reasonable sampling algorithm. An unfortunate thing is, if you notice the negative phase of the gradient of the energy-based model or the log likelihood term, in fact, tells us that we need to be able to sample from the model that is being trained. So if we cannot sample well, we cannot learn this model well. If we cannot learn this model well, we probably cannot sample from the correct distribution. So we run into a vicious cycle. And it's a pretty horrible thing that happens. And this is my attempt at trying to draw an intuitive picture of why that is a horrible thing, but I don't think it's intuitive at all. So you can just trust me that because we have to sample from this model in order to train this model, if you cannot sample from it, yes, training is not going to work well. If the training is not going well, sampling can also be even more challenging. So you can just trust me on this. And indeed, between 2006 and 2012 or so, the whole large portion of deep learning or the deep learning research has been on solving this issue. Already starting from the 2002, Jeff Hinton talked about the contrastive divergence, which was extended over the next many following years into persistent contrastive divergence, fast persistent contrastive divergence. I think there were, let's say, a couple more as well. And then you had some of the people in Montreal as well as in Finland, we worked a bit on the parallel tempering or adaptive simulated tempering, so essentially introducing parallel version of the models with the different temperatures in order to facilitate sampling. But none of them actually worked, unfortunately. And it's still an ongoing scientific endeavor. If you were impressed by, just like I was, on the diffusion-based models these days, like the DALI 2, or what was it? There was another one from Google, right? Not the PALM. Imogen, and so on. Those are another, let's say, line of work that tries to tackle this challenge of training a generative model. And then that is based on the diffusion-based generative model from the Joshua in 2015, and then has been pushed very further. Now, there are many studies, recent studies that show the impact or the effectiveness of the flow-based models. And of course, there's been let's say, huge interest in generative adversarial networks until a couple of years ago as well. So then let's just think about why it's so difficult. So manifold assumption tells us that the high dimensional data lie roughly on a low dimensional manifold. And then there is a blessing. There is a blessing that allows us to build this kind of this gigantic neural net on a gigantic space without too much of an issue these days. But then when we try to fit a generative model, this becomes a curse. Why is that? In order to identify or characterize this low dimensional data manifold, our generative model should look at high dimensional non-data manifold and then trying to suppress the probabilities that are being assigned there. And then that's just very difficult. The reason why machine learning works well on high dimensional space is because data lies on a low dimensional manifold. However, for us to build a generative model, we have to explore the entire space and then suppress all the possible values that the input can take. And that's just too big. Now, what does it mean for the space to be high dimensional? When we talk about the, discrete combinatorial space, let's just say that the number of potential, let's say a value that input can take is just gigantic. I'll just say it like that for now. So instead, it turned out that already from 2002 or even earlier, we've known what we should do. And what we should do is to simply ignore all the points in the space that are just far away from the data. And then that's the kind of, let's say, foundation behind the contrastive divergence. So instead of looking at the all possible space, we always start from one of the training examples, and then training examples are by construction or by definition on the data manifold. And we want to just look at a small neighborhood. And then within the neighborhood, we're trying to ensure that the energy values are low on the training set, training data points, but not or high on all the other parts of the neighborhood. Again, some nice looking intuitive picture it was supposed to be, but it doesn't look like so. But what you can what you should see from this one is essentially that anything that is far away from this, let's say, curve are being ignored. We don't care. We're saying that, well, our learning algorithm is having a difficult time trying to look at the entire space. So we're going to relieve our learning algorithm from having to explore too far away from the data. So what is the advantage of doing so is that as long as we start sampling from this model start sampling from the model starting from the data manifold, and as long as we know how to stay nearby, then we can actually sample a lot of, let's say, realistic looking examples, even if we train our generative model to focus only on the neighborhood of the data manifold. Now, the downside is that of course, if we continue to sample further and further, it's going to eventually drift away and then find those spurious, let's say, modes that are very far away from the data manifold. So we just have to know when to stop sampling. And starting from this perspective, what you see is that we actually don't need to build a sophisticated generative model that gives us the actual log probability or the energy value. All we need to know is how to denoise a corrupted version of data. Why is that? The data points that is clean is the point that is on the data manifold. And then the data point that has been corrupted with some kind of noisy process is the data point that has been kicked off the data manifold. If I know how to bring that back onto the data manifold that is doing the denoising, then I should be able to start from some point on the data manifold, kick it off the manifold, bring it back to the manifold, kick it off the manifold, bring it back to the manifold, and then thereby I can actually sample from this entire space of the data manifold quite efficiently without having to worry about going too far away from the data manifold. And so denoising is sampling, but then the question is, okay, what kind of corruption do we need to work with? And then what kind of, let's say, denoising function are we going to work with? And it turned out that of course, denoising, we just use a neural network, like the gigantic neural network that you have already learned. And this morning you learned about transformers. So if you're working with some kind of sequential data with the discrete, let's say, tokens, you just plug in the transformer. And then this transformer is going to take as the input a corrupted version of the input that is slightly off the data manifold. And then it's going to just push it back onto the data manifold, and we train it as a denoising autoencoder. And what should this corruption function be? This corruption function should be as unstructured as possible. So every possible ways in which the data point can be kicked off the data manifold should be included so that this denoising autoencoder can figure out what are the valid directions and then what are the invalid directions. So once we do that, all we need to do is just get the unstructured corruption distribution. And then this one doesn't have to be learned. We just decide. And then we just train a transformer to reconstruct. And we do it over and over the entire data set with the multiple sweeps because this corruption function is going to be stochastic. And then once training is done, all we need to do is just ensure that we start from the nearby point of over the data manifold, because otherwise this will still create all these spurious modes that are far away from the data manifold. And then the sampling procedure can get stuck in these spurious modes. So one thing that I want to quickly point out is that just because of my drawing and our limitation in our imagination, it feels like things can only move really locally. So we had some local noise, we tried to denoise it, local noise, denoise it. But it turned out that that's not necessarily true because our denoising part can be highly, highly nonlinear. And also the noise that we add does not necessarily keep the point in a local neighborhood. And at the end of the day, what does it even mean for the local neighbor to be in this kind of, let's say, combinatorial space? So it turned out training with this kind of local noise and training this denoising autoencoder allows us to explore the space quite dramatically. I'm going to show you some of the examples later in the context of proteins. So this is the first part of the foundations behind building a deep learning system for protein design. The first part, that is the generative model. And then because we're working with the discrete combinatorial variables, we actually do use a denoising autoencoder rather than all those fancy, like the diffusion-based models and whatnot. Although diffusion-based models, if you're familiar with it, is exactly a denoising autoencoder, but with the multiple steps. Is there any questions before we go into a slightly more boring, let's say, supervised learning part? I don't know why everyone is so excited by supervised learning, right? Yes. But anyway, if you have any questions, you can always interrupt me. And otherwise, I'm going to go into classification as well as a regression, because unsupervised learning is all fun and amazing to the point that we can train and then sample amazing looking stuff. But if we cannot guide the sampling procedure from an unsupervised learner, it's a bit of a, a hobby than anything serious. So we got to make it really serious. And serious is, in fact, the discriminative or the supervised learning here. And then we talk about the conditional modeling where the input is going to be X and then target is going to be output. So we're trying to learn the distribution over this output, the Y, given the X. And in this particular case, we're going to largely focus on what I'm going to refer to as unstructured Y. That is, the output is going to be essentially extremely simple. It's going to be either one of the k possible classes, or in the case of the regression, it's going to be extremely low dimensional. And then the correlations among these dimensions will be low. Now, it doesn't have to be like this. We can always make a conditional generative model to capture all those complicated statistical dependencies among the dimensions of Y, but that's kind of, let's say, out of the scope of this talk. And discriminative modeling or the supervised learning is where the actual success of deep learning has been so far, and in particular in drug discovery as well. In molecular design, so for instance, in the small molecule design, which kind of aims at making those pills that you take, what happens is that we often have a gigantic library of possible small molecules, or the small, let's say, constituents that we can actually mix and match to build a tons of, let's say, candidates. And then the goal is, given a new disease, which one of these, let's say, 10 to the often, let's say, power of 8, but I think we can go to 9 and 10 these. 9 and 10 of the candidates are going to be worth testing further and further. So we have this, the extremely quick, rapidly shrinking funnel. And then doing so takes a lot of time because before it was all about experiments. And when I say experiments, it's not, let's, start up, let's say, I don't know, like the SSH, the terminal, and then, let's just, get use the Slurm to launch some jobs. It's the actual experiments where we do the in vitro, let's say, experiments, and eventually we need to do the animal testing and human trial and so on, right? So it's extremely complicated and expensive. So the supervised learner has been used here and there in order to speed up some of these stages in this, let's say, exponentially shrinking funnel. And it's been quite successful, although it's very new in a sense that we actually haven't had any real drug that was selected based on this kind of supervised learning based, let's say, filtering yet. And that also happens with the larger molecules like the proteins as well. And then in this particular case, we've been using them for the purpose of the computational design in particular. But I'll talk about it a bit later. And one thing you realize at some point is that any kind of classifier you train, in fact, defines an energy function on its own. So we have a conditional model that computes the probability of Y given X. But then let's say I have some target value Y. So I want to know what kind of input corresponds to the target value Y had. And then all I need to do is I just look at the value that is assigned to my classifier or the regressor on the particular Y hat. And then that itself already defines an energy function over the input space. Now we need a bit of a regularizer. I'm going to talk about why we need them. Unfortunately, it's very difficult to define what that regularizer should be without knowing about the target domain. So in other words, if we have a classifier and a target class, we can define a distribution over the input according to how probable that class is given an input. And this is, of course, not a new idea. We've been known about this for many years. Already in 2014, Karen Simonian, who is now at DeepMind, but was back then at Oxford, realized that, if we have a deep convolutional network that has been trained really well, now we can just pick the class, one of the class that I want to inspect this model about, And I'm going to do the let's say optimization to find the input, the X, that's going to have the highest log probability assigned to the particular class. And then these were some of the nice, let's say, visualization he had. Nowadays, if you look at them, they don't look at all like anything realistic or nice. But back then, it looked really amazing. Like if you look at the bell pepper, you actually do see a couple of the bell peppers there. When you look at the husky, if you look at it really, really carefully, you're going to see a bit of a husky near the center. Do you see them? Just try a bit harder, you'll see them. Yeah. But then, I think one of the reasons why it didn't look at all like was identified by the team from Google back then in 2015 as the fact that this energy function is not really constrained enough. So what they did was to design a regularizer, R of X, really carefully, so as to impose some of the knowledge about the domain that we know, and in this particular case, it's going to images. And then they wrote a blog posting, and then it was really like amazingly better in terms of what we are actually getting out of. And then it kind of made sense. Like if you look at the bananas, you do see bananas there. And if you look at the screws, you do see tons of screws. Now, it also revealed the problems of some of these supervised learners that we've been using and training. If you look at the parachute example, what you see is that you do see parachutes, but you'll also see that there is a person who is in fact riding a parachute. And then if you want to build a parachute detector, and then let's say you create a startup based on the automated parachute detection, and then you want to sell it to the parachute, let's say manufacturer, your detector should not look at a person and then say that there is a parachute. On the other hand, these classifiers, because of the reasonable, let's say, bias that you see in the training set, where the parachutes are almost always being, let's say, ridden by a person, it will actually learn to see that, oh, here's a sky and a person that is falling down. That person probably has a parachute, because you usually don't add in people just jumping off the plane without a parachute in the data set. So, it was very, informative generally. But still, they didn't look really well. So in order to talk about this, we have to talk about, how is this classifier doing this, right? So it turned out we thought we were training a classifier, but we were in fact training all these generative models ourselves. And the reason is because any powerful classifier has to memorize training examples. And one of the representative examples that you have probably already learned earlier this week is the k-nearest neighbor classifier. K-nearest neighbor classifier, if you know how to choose the distance function really well, is extremely powerful. In fact, you set the K to 1 and then have many examples and so on. It's an optimal classifier to use. And then this powerful classifier by construction memorizes every single training examples there is. And in this case, we actually choose a proximity or the notion of proximity. But then we can now think about other similar types of the classifiers, such as the kernel machines. Kernel machines, we save almost every training examples, depending on what kind of regularizer we are going to put. And then we say that proximity, too complicated. We're going to simply just send the input into the infinite dimensional space and then let everything solve there. So using just a random projection. Or of course, we can use deep learning where we're going to learn the notion of the proximity from the data directly. But in all these cases, most of these classifiers will have to memorize a large chunk of the training examples. And that's how these classifiers, in fact, become a generative models as well. Unfortunately, one thing that we don't do with the discriminative modeling as opposed to generative modeling is to suppress the energy values or to suppress the log probabilities assigned to all the configuration that do not look at all like the training set. And then that is not what we do here. And then that is the reason why when we did the visualization using the classifier's output as an energy function, we saw something that looked really reasonable, but none of those actually look like the actual training examples that we use to train these large scale models. In other words, there's no constraint outside data nor between training examples. And this is a really nice figure from Jason Yosinski and others' paper from 2015. So you can imagine this kind of, let's say, two-dimensional plot. So x-axis is going to correspond to the input space, so image space in this case. Y-axis is going to be our imaginary, let's say, axis or the axis that tells us about whether that image is recognizable or not. And what you see is that the training examples are all at the very top. And then we use the training examples there to fit our classifier. But then this classifier can, in fact, work with any points that are unrecognizable as well. And then just by the virtue of training as a supervised learner, these classifiers are going to put a high-low probabilities or the low energy on these unrecognizable points as well. And then that's what we actually already saw with the earlier examples of the visualization of these deep convolutional networks. So what we mean is, what we need is something beyond a single classifier. There are just too many holes and empty spaces in this space, so we want to put more constraints by using now the generative models that we talked about. If X, the input were continuous, everything is so beautiful. In fact, already in 2016 or 2017, Anand Nguyen, who is now at Alabama or somewhere, he did a great, actually, he demonstrated a great, demonstrate this capability of combining generative model and the discriminative model for the purpose of sampling from a classifier. What he did was to start from a classifier that has been trained. Oh my God, that figure is the most important figure, but somehow I'm actually blocking it. But all right, let's imagine that. Can I move it around? Oh, I should move it around from here. Okay. Okay, that's important. Yes. Now you see the important plot there. So C is a classifier that's going to take as the input X and then outputs one of the probabilities over all possible classes. We're going to pick one class, the red one, and then we want to sample from the energy function defined by this classifier. However, we want it to be constrained to lie on the data manifold, and how we impose that constraint is to plug in the denoising autoencoder that we already talked about in the first half of our talk, so that we never actually go too far away from the data manifold. Denoising autoencoder is going to always push it back toward the data manifold as we're trying to change the value of this according to this classifier. That turned out to be simply to start from some point X and then do the gradient descent, or the gradient descent with some noise, where the gradient gradient comes from both the generative model, the denoising autoencoder, and the discriminative model that is a classifier. And by doing so in 2017, An showed these visualizations, and that was kind of revelation to a lot of people, including myself. Now you could actually see that these are images. The red shank, you see the birds there, and you see the ants there. The number of the legs, is not perfect, often too many of them, but still you do see ants there. Monastery, it's like perfect, almost perfect, right? It's a bit Escher-like, but still you see the monastery there. So what that means is that only if we can actually use this for a discrete combinatorial input, we might be able to get something like this, and then combine generative model with the discriminative model for the purpose of design. And then that's what we call deep manifold sampler. So we had this at the very right before we formed the company. So in 2020, mid 2020, we had this technology, and then we had to decide whether we were going to write another NIH grant proposal. By the way, not fun at all. So I highly recommend you against it, or go talk to the venture capitalists and then try to ask for their money. Turned out that the NIH is actually more difficult to get money out of than these VCs. So we decided to go with the second route. So I'll just show you how we actually combine these two ideas into building a computational protein design model. So one thing is that whenever you run into this kind of new problem domain, you need to think about what are the kind of, let's say, high level but important criteria that your algorithm must satisfy. And they tend to differ from one domain to another. So in the case of this kind of computational protein or the computational molecular design, one thing that we wanted to ensure was the fact that the sampling had to be iterative. What do I mean by that? Is that we cannot simply ask our model, get me one nice design of protein. We can't really do that. We in fact want to start from somewhere that we know is reasonable. And then we want our model to propose a candidate changes. And we do it over and over until we find something that is good. The reason why we need that is because we need to be able to easily incorporate external oracle, computational or the biophysical oracles to guide sampling. And in other words, we need to be able to intervene on the process of this sampling ourselves. And then the second thing is that we need to be able to control the degree of exploration. Often when you think about the generative models, and in particular, if you've been playing around with the DALL-E 2 and GPT-3 and so on, we tend to get one good answer, or perhaps, I don't know, 2 to 5 very good answers. So we try to maximize the low probability, or to find the samples that maximize the low probability. But in this kind of design, that's exactly the opposite of what we want to do, because our goal is to find something that nature hasn't figured out yet. What that means is that if we start with all the data that we have that were produced by the nature and biology, and then train a model, and then trying to find the one that is extremely high probability, then that's exactly probably what nature and evolution will eventually find themselves. And that's not what we want. We want to be able to go beyond what they want. So this is really important. Second one is that the length must evolve during sampling. Now you've been listening to all these amazing NLP researchers, so you think, that's so natural, right? Of course, length has to change. But when you talk with the other people who actually haven't worked with the natural language or audio or whatnot, to them, everything has to be fixed. In particular, because for people who actually have been working with the Theano and TensorFlow, changing length is just horrible sin. It's very difficult. But then, of course, in the case of the biology, and in particular for the proteins as well as the small molecules, the length or the size has to evolve during the process of sampling. Not only we cannot even just decide one length and then just sample, but the length has to change over time, because some of the interventions that we're going to do may introduce some of the, let's say, tokens, or remove some chunk of the tokens in it. And furthermore, this actually enables some non-trivial objectives such as compression. What do you mean by compression? What if we have the gigantic protein that does amazing stuff? Can we actually keep this function as it is, and then try to just shorten it little by little? And then that makes it really different in a sense that the delivery is going to be easier. And then they might be able to, they might be flushed out much more easily. And then we'll be able to target much more, let's say, organs as well as the different types of the cells, because they're going to be smaller. So, this actually allows us to work with some non-trivial objectives. And then the third thing that is a bit different from many of the things that you have seen so far is that the diversity is super important. One thing I can tell you with like 100% confidence now is that the real biology, real chemistry, real physics, they don't live inside our computers. Real biology lives inside us. Real biology lives inside rabbits, rats, llamas, monkeys, but they are definitely not living inside our computer. What that means is that the experiments have the final say on the quality of designs. So we cannot really simply maximize the quality that is defined or determined by our computational models. So we have to prioritize the diversity as the most important criterion. So with this in mind, let's just put some of the things together. So the ingredient one, a sequence denoising autoencoder. We actually used it in 2016 for some random purpose with the Felix Hill, who is also at DeepMind. Somehow everyone is at DeepMind, I don't get it, but anyway. And then now we decided to use the sequence denoising autoencoder. However, some tweaks. One is that, of course, we are now living in the era of attention and transformers, so we're going to use multi-vector representation. So we're not going to compress the entire sequence into a single vector. Second thing is the length transformation. We want to be able to change the length on the fly. And then for that one, we can use various techniques. We use one particular technique from last year's paper on the non-autoregressive machine translation. And thirdly, because we wanted to maximize the diversity, and then one thing that we haven't been too successful is to maximize and control the diversity, the level of the diversity of the autoregressive model. So we just stick to the non-autoregressive model. But on top, we actually use CRF in order to at least ensure some of the local consistency. I heard that this year's summer school is not teaching CRF, but CRF is actually very useful. I can tell you. Yes. So if they check out the last year's or the year before's video, I'm pretty sure there is a lecture on the CRF, right? Last year. Yes. Okay. So very, very useful. Very useful. So non-autoregressive decoding CRF on top, that actually makes a lot of things much better. And then second ingredient is a function predictor, because as I said earlier, just sampling nice looking protein sequences don't really get us anywhere. It has to be extremely, extremely targeted. But unfortunate thing is that the discriminator that we're going to build, if we build it on top of the actual protein sequence, is not going to work, because there is no gradient of the discriminator's output with respect to the input. Input is just discrete, and even the size changes. I'm not even sure what it means for us to compute the gradient there. So instead, what we do is we actually plug this classifier on top of the generative model, sequence denoising autoencoder we have. So we train a sequence denoising autoencoder first on a large amount of the unlabeled examples. Once training is done, right before the length transformation, we plug in the classifier on top of that to predict the functions. And then now, because in the middle, everything is continuous and okay behaving, so we can now compute the gradient of the output of this classifier with respect to the intermediate representation of this denoising sequence autoencoder. And then once we plug that in, we just simply encode the input into a vectors, compute the gradient, add the gradient slightly, and then we decode it out. And then we do it over and over in order to get a series of the samples. So this is how it looks like. We start from top. So in the case of the proteins, you can imagine a single protein as literally a sequence of letters where each letter is one of 20 or 22 if we start counting the non-canonical amino acids. And then we apply some corruption that may change the length. So we're going to drop some of them, we're going to add some of them. And then we feed that corrupted version into a transformer encoder. And then we have the length predictor that's going to predict the length difference between the original sequence and the corrupted sequence. And according to the predicted length, we're going to do the length transformation. So we're going to change the number of the vectors. It's just often a simple upscaling or the downscaling, or sometimes we can have another transformer for the length transformation as well. And then before we feed it into the transformer decoder, we're going to send the vectors to the function predictor, which is in our case, again, a bit of a mix of the convolution and the transformer to predict the property. And then whatever is coming out of the length transformation also goes into the transformer decoder during training. So we actually train the transformer, so the denoising autoencoder and the function predictor separately from each other. So we're not going to backpropagate the gradient coming out of function predictor into the length transformer nor the transformer encoder. Once training is done, let's focus on the dotted, let's say a pink line curve. So we feed it in, and then we're going to send it to the function predictor. We compute the gradient of the loss according to the target property we want. Then we're going to change it. We're going to perturb it a bit according to the gradient, and then we decode it out. And then that's how we do the one step of sampling. So I'll show you some examples. Is there any computational biologists or the biologists here? No, okay, so I can really literally say whatever I want then. Very good, yes. This is a bit of a small scale experiments, and it's not on antibodies, it's on just generic proteins. So we train on 20 million unannotated sequences of the proteins from the PFAM database, the sequence denoising autoencoder. And then we use 500,000 annotated sequences from the UniProt database in order to predict the functions. Some of the functions are pretty high level as well. And this classifier is built on top of this sequence denoising autoencoder's intermediate representation. So first thing we wanted to check is that even without guidance, if we just do the sampling for a few steps, will we actually stay on the data manifold? Now, one unfortunate thing, by just looking at the sequence of the amino acids, except for a few important bits, we can't really tell whether the protein is a good protein or not. Sometimes that is not even a protein. It's very possible. It's unlike natural language where we're going to read and then see, well, it looks pretty solid or not. We can't really do that in this case. So what we did was to use, okay, I'll just move this. Oh, that's a good point. Yes. So we started from one protein, so that is marked as a wild type, and then we use a computational method to estimate the rough energy level of that wild type protein. And then what we did was that starting from there, we evolved it or did the multiple sampling iterations up to about 4 to 5, and then for each one of them, we're going to look at how far it is from the original wild type protein in terms of the edit distances. So the number of residue changes there. And then we look at the energy again, computational energy. And then the energy levels of all these generated sequences do stay lower than the wild type's energy. What that means is that it's not anything too crazy. So it does seem to actually stay on top of the data manifold. And from there, we decided to go for the next small, let's say, experiment where we start from the kind of, let's say, metal binding protein. So if you see that small, let's say, circle there, so there's going to be a metal, and then there is a small pocket that this protein can actually capture the metal. And then this is a known the existing protein. And then we remove that binding part. We remove the binding site by just removing some of the residues, the red colored ones. And then what we did was, starting from there, we sampled from our deep manifold sampler while telling it to maximize the chance of exhibiting the function of calcium ion binding. Calcium ion is another metal. So then after a few generations, what we see is that this pocket was reintroduced automatically. Although we didn't actually tell it exactly where and how it should be reintroduced. This actually does tell us that we can indeed guide the process of sampling based on this classifier alone. And then we did another experiments, this time with I think the cutinase activity, which is kind of important if you want to eventually build enzymes that are going to consume the plastics or something like this. So we saw something similar. And then we could even change how these proteins fold dramatically by just working on the sequences. We could in fact change how the folded structure would be after the folding happens. And this is kind of a nice example. The example that I like, because we never use the actual protein structure here. We only use the primary structure that is sequence. And you probably have heard about the AlphaFold 2 and whatnot, where they predict to a almost atom level, the precision, how the protein is going to fold in a neutral setup. So that's an amazing fit, but it turned out that that is just one side of the story. And for the purpose of the design, knowing structure helps, but that is not necessarily the main driver of how we design the new proteins. Although I think the AlphaFold 2 is super cool. Yes. So yes, so I'm about to wrap up. So the generative modeling means discriminative modeling. So in fact, doing both of them turned out to be extremely, extremely important. And then by doing so, we can really speed up design, because generative modeling narrows down the search space intelligently and dramatically. And then within this narrowed down search space, discriminative modeling or supervised learner can actually tell us which subset that we need to choose from. And then obviously there are many different ways to combine these two paradigms. The deep manifold sampler that I just presented today is one way to do so, but there are many different things like GFlowNet that which Joshua is completely obsessed with at the moment. But it looks interesting. And then there are many different ways to go about. You probably have seen all those classifier guided, the clip-like models and so on. But of course, at the end of the day, every problem requires different types of the criteria. So what is the right way to choose a right, let's say, algorithm, combine them and improve them is going to be up to, let's say, you who is going to actually solve the problem. Obviously, this is not really end, but the very beginning. So we use the very same technology for antibody design. And then we know that quite few of them are actually going to express. We run the experiments internally ourselves, and then we know that some of them actually do bind to the target proteins or the molecules that we want them to. However, we are still in a very, very primitive, let's say, stage. So two things that we really need to work on at the moment is, one is the uncertainty quantification plus the active learning, because it really helps us maximize learning and also gain trust in our own designs. And then uncertainty has to be defined not only in terms of the property prediction, but also in terms of the sampling. So can we actually quantify the uncertainty in this combinatorial space? I don't think anybody actually has a good answer to that one. And second thing that we are looking into is the multi-objective sampling. So when we think about the multi-objective optimization, you often immediately think about the black box optimization with the, all those finding the Pareto frontier in this kind of low dimensional space. And then you read some of the papers, they're going to say unprecedented scale or the multi-objective optimization, unprecedented scale of the 32 dimensions. We can't really use that kind of algorithm because we're working in 1,000, if not tens of thousands of dimensions. So we need some efficient, effective, let's say, way to explore this kind of trade-off across all those different objectives, perhaps using some kind of gradient information. And well, okay, so one of the actual figures don't show up, but at the end of the day, what we're building is a lab-in-the-loop design platform that has a computational aspect and also the wet lab aspect. And then what is the right way to have those two components interact with each other in order to design a new de novo proteins or the molecules is a big challenge that we're trying to solve. And then all the things that you have learned this week, except for CRF, are very critical to building such a platform. And that actually concludes my talk. Thank you.
So you have to start the I understand this is a sort of classifier guided generative.
Yes.
That actually precedes this famous case of GAN slash classifier. So it's kind of earlier.
Yeah, so we actually had this one from about 2019. Just at the Unity, we were, well, first of all, pandemic started, so we were a bit lost, and then everyone was burnt out. And because Unity, we were thinking quite a lot of. So the one thing that makes it a bit challenging for us to publish in this domain is it cannot be done as freely as the machine learning papers, because there are two ways to go about. One is that we can blindly say that these computational tools that people have been using are so awesome. We're going to just trust that if this, whenever these computational tools tell us that our designs are good. But one thing for sure is that these computational tools are not perfect. So if you want to really write a paper with the real validation of the technology, we actually have to run the experiments. And then that can be a bit tricky. So that's why it's taken us so long. And then even now, we actually haven't been able to publish anything that is based on the real experiments yet, because we're still running the experiments at Generative now. We do have some of the workshop papers and a few papers on a completely, let's say, method-oriented ones. But there, we always have to put some kind of disclaimer saying that, we actually haven't tested these designs in experiments extensively yet.
And then a follow-up. So in the meanwhile, the diffusion models have kind of overtaken classifier guided universe. Do you see something like diffusion working or protein sequences? Again, caveat of discrete variable.
Yeah. So I mean, the diffusion-based models are, in a sense, as I mentioned earlier, diffusion-based models are a denoising autoencoders, but they are kind of chained. So we're looking at multiple steps, right? And then in order to speed that up, we often rely on some kind of differential equation, and then fast solver in order to quickly, let's say, diffuse and then reverse diffuse. In the case of the discrete, you can in fact come up with a diffusion process for the discrete space and also trying to reverse it. That's definitely possible. I think there was a paper earlier this year or the late last year on the discrete diffusion process. But there, what effectively happens there is that because there's no gradient that is actually passing through those multiple diffusion steps. So I'm not entirely sure whether that should be any different from just training a denoising autoencoder with potentially just more than one steps of the corruption and denoising.
Thank you for the talk. I'm curious about, so from the classical machine learning point of view, this combination of autoencoder from and you put the classifier on top of the space seems like this the model, the unsupervised model, transform the input data where the future map can be separable was a very easy classifier, I would say. And this scheme reminds me the current NLP the approach where we pre-train language model and fine-tune it on any task that we want to solve. I was wondering, can we have any interpretation where we can see this language model pre-training and fine-tuning from the perspective of this data manifold, data representation?
Yeah, so that would have been the kind of remaining question part of the other potential candidate talk that I was going to give here. So I was like, this or the data augmentation, but you're perfectly correct in a sense that these language models that we pre-train and then fine tune in order to build a better classifier or the classifier that generalizes better with a few examples. This practice does connect to this kind of capturing the data manifold and whatnot. However, it's not clear whether that is the only thing that is being done. Right? So maybe there are many different ways to pre-train these large-scale models and fine-tune it to get the similar, let's say, type of the accuracy. In which case, we'll have to figure out that what is actually going on there. But that said, so coming back to the very early point, so people indeed have been using the unsupervised learning as a way to, let's say, transform the input. So for instance, it used to be quite a widely used practice of using PCA in order to reduce the dimensionality before, you fit the classifier on top, or, using some other kind of clustering in order to get a different representation. And until, right before the deep convolutional net took over in 2012, one of the most successful, let's say, feature extraction technology was using a kind of, what was the name? A Fisher something. They just, dramatically increase the dimensionality, and they use a bit of a clustering there as well. But then often the reason why they were doing, in particular, where they reduced the dimensionality, was because they were just scared. Because our learning theory tells us that if the input space dimension is large, we will require a lot of examples. So they got scared, and then they were like, oh, can we actually reduce the dimensionality? Then we're probably going to get a better generalization or something. But nowadays we are not really scared of the high dimensional space or the large scale models or the models where we have absolutely no guarantee. I mean, no one said it as if it's a bad thing, but I think just being brave is always better than being timid. So we are not scared of them. So we can actually now have a slightly different perspective onto what is the right way to combine these generative models and discriminative models.
Hi, and thank you so much. It was super interesting. I wanted to ask some of the things here looked a bit like things I know from reinforcement learning, but I'm not an expert at all of it. And I wanted to ask your opinion on it or about reinforcement learning in general, and if you can use it in the context of generation sampling.
Yeah, for that question, okay, I think that you need to just, is Chris around? You need to find Chris and ask him, because I think Chris and I think that Wen was on that paper as well. They put a paper with an algorithm called MED or something like that, like a week ago or so. I think that that's the only algorithm where the reinforcement learning or any kind of policy gradient has been used to train this kind of language generation system, like the machine translation system, to actually increase the reward function that you designated. So you definitely want to talk with him. We do look into the reinforcement learning a bit, but one of the challenges here is that we don't really have a good reward function. So these function predictors that we use are extremely noisy because the experiments are noisy. So for that, I'm not entirely sure whether we can rely on this kind of function predictor that much. However, in the lapping the loop scheme, so instead of thinking about the traditional reinforcement learning where we have the immediate response in terms of the action and reward, we do look at the some of the techniques from the offline reinforcement learning, as well as a batch bandit type of thing where we have to look at, we send out multiple set of actions, and then there's going to be a gigantic delay of, let's say, 2 months until we know which ones are good. So we're definitely looking into that. Not too successful. So.
Hello, thanks for the great talk. I was wondering if you could elaborate on how you ensure or how you guarantee diversity in something.
Yeah, how we, yeah, I mean, the one way to, yeah, so it's going to be a really weird answer, and I think it's a very imperfect answer, is that by making our decoder less capable of capturing the very strong local dependencies across all those residues, that's actually one way for us to ensure that there is some level of diversity. Now, of course, then what is the best way to ensure the diversity is, as you learned yesterday, we're going to simply say that every token in the protein sequence is going to be independent of each other. We sample from it, we get huge amount of diversity, but most of them are going to be pretty much useless. So we now use a transformer decoder that's going to be able to kind of, let's say, reduce the space so that it can generate something that is more reasonable. But again, we use a non-autoregressive model where we, again, assumes the statistical independence between all these tokens, given the previous, let's say, corrupted version. So that one turned out to be still not good enough. So it's diverse, but not good enough. So that's why we put the CRF on top of that. And then CRF with a very simple, let's say, even the bigram, let's say, capturing the bigram dependency, it turned out that that's already good enough for us to get a pretty high fidelity sequences, but with a high level of diversity. And we are still looking into various other ways to ensure that we keep the level of the diversity while increasing the fidelity of these designs. Some Latin variable model, I guess. Okay, I heard that it's time to have some dinner. So, yeah. Thank you.